Hello, and welcome to my lesson on G protein couple receptors. I am Teacher Joseph. I'll be covering the basics of what they are, as well as some of the mechanisms of what they do within the human body. Now, at first glance, this might be a little overwhelming. So many different steps and potential directions to go within this single pathway, commonly referred to as the GPCR pathway. But you might be asking, what is the point of learning this? What does this pathway do? And all I have to say to that is, patience. Patience, please. That's all I ask. And all shall be revealed. Okay. So now, in order to really understand anything, we have to get a little bit into that background. So, let's dive in with 2 feet and begin with the question of what is a receptor. And then we can go on from there. Sounds like a plan, right? Perfect. In the system of chemical communications that coordinates the function of all the different cells in the body, receptors are the sensing elements. The chemical messengers can be hormones, transmitters, as well as many other potential mediators. As you can see in the graphic, there are 4 receptors covered, and each of them are covered quite thoroughly, in my opinion. And they include even the timescales and examples. So let's just take a second and look at those. So in the first one, we have the ligand-gated ion channel, or the ionotropic or tropic, some people like to say otherwise, receptors. Whereas you can see here, the timescale is milliseconds, by far the quickest of all the receptors in this diagram. And some examples are ACH receptor. While our second one, which is going to be the most important of them all for today, is the G protein coupled receptor, or the metabotropic receptor. And as you can see here, instead of milliseconds, it is seconds, with an example being the ACH receptor, but instead of the nicotinic, it'll be the muscarinic. My apologies. I should have made it a little bit bigger for everyone to see. Leaves us 2 more to go, the kinase-linked receptors, and the nuclear receptors, which both of them take hours, but they're examples. Obviously, they're not the same thing. The example for the kinase-linked receptor would be a cytokine receptor, as opposed to the nuclear receptor being an oestrogen receptor, or oestrogen. Yeah, that sounded a little funny. Anyway, as I gave a little hint to, and the title of our lesson for today, the focus for this presentation is none other than the second receptor, the G protein coupled. So, okay, that takes us now to the G protein coupled receptors. So the G protein coupled receptor, or if you want to shorten it, GPCR, have many different names that you could use. One being 7-transmembrane domain receptor, and the shorthand of that, the 7TM receptor. They also have a name serpentine receptor, and they also have G protein linked receptors, which is similar in name, just instead of the C for coupled, it's linked, so GPLR. and they constitute a large protein family of receptors that sense molecules outside the cell and activate inside signal transduction pathways, which, as we saw in the prior diagram, we could the end result being some kind of cellular response. And that might, and some of those names might bring the question of like, where do they derive? And probably the most interesting of all, I would say, would be the 7-transmembrane receptor, because it's the most descriptive of them all. And that is due to the fact that these receptors pass through the cell membrane 7 times. So 7 transmembrane receptors. Trans meaning from inside it out throughout the membrane, and there are 7. So can't really say much more about that fact. So in general, since GPCRs vary in size and function, they can also further be categorized into families. But of course, they are going to be determinate on these attributes. And of course, as you can see here, some of those size differences are going to be uh, the small molecules, peptides, large proteins. Incredible. Very good. As promised, we're now going to dive into a little bit of the families of the GPCR. And as you can see in this diagram over here, let's just highlight that. This diagram right over here, you can find the families that we'll be covering in this slide. And they are color-coded accordingly. So let's just take that quickly away. Don't want to distract anyone. Perfect. Very nice. So according to the basis of phylogenetic criteria, human GPCRs can be divided into 5 main families, which include glutamate, or as you see here, also called metabotropic glutamate, rhodopsin, adhesion, frizzled taste 2, and secretin. My apologies. Glucagon receptor family is another way to label the secretin as well. And the most prominent or the most well studied of these 5 is the rhodopsin, of course. And the second one, which has 33 different members, is the adhesion. Okay, so let's just quickly go through how that is. So I'm just going to circle around. So this area right here, I know it looks pretty big. Unfortunately, I didn't make it all the way, but all this blue area right here, that is part of the rhodopsin family. So as you can see how vast the roots in this tree, so to speak, are. As opposed to right here, we have the adhesion. So still quite plentiful, but nowhere near as much as the rhodopsin. And going down the line, we then have the secretin here, which is pretty close to the adherent, but still noticeably smaller, not as diverse. Then we have the glutamate, or the metabotropic glutamate. And finally, with the green is our fizzled and taste 2. Right there. Perfect. Awesome. So our GPCRs interact with a number of ligands ranging from photons, ions, amino acids, pheromones, neurotransmitters, peptides, proteins, hormones. But there's also still a category called the orphan GPCRs, in which the natural ligand needs to still be identified. And with that being said, let's just dive a little bit deeper, or at least look a little deeper into a few of the families that we just covered. Specifically, we're going to start with the most plentiful of them all, at least according to this diagram, the rhodopsin family. Okay, wonderful. Rhodopsin receptor family. So the rhodopsin receptor family, or shorthand RLRs, are a family of proteins that comprise of G protein coupled receptors, of course, and are extremely sensitive to light. It activates the G protein transduction, or if you want to shorten it again, GT, to activate the visual phototransduction pathway. Mutation of the rhodopsin gene is a major contributor to various retinopathies. Remaining receptors are liganded by known endogenous compounds. Examples of these include FXR or farnesoid X receptor, which is activated by bile acid. We also have liver X receptor, shorthanded LXR, and peroxisome proliferator activated receptor, or PPAR. Perfect. So if we don't have any questions with that, then let's continue on with our next two. The next two families that we'll be covering now is secretin receptor family and the metabotropic glutamate family, or simply glutamate family. Starting with the secretin receptor family, it is a family of GPCRs that includes razoactive, intestinal peptide receptors, and receptors for secretin, calcitonin, and parathyroid hormone, as well as parathyroid hormone-related peptides. These receptors activate adenylyl cyclase and the phosphatidylinositol calcium pathway. Wow, that's a mouthful. As opposed to the glutamate family, the metabotropic glutamate receptors, or you want to shorten those a little, because that's a little bit of a mouthful as well, MGLURs, are family C GPCR that participate in the modulation of synaptic transmission and neuronal excitability throughout the central nervous system, with high levels in the cerebellum and thalamus. They have been subdivided into three groups based on intracellular signaling. And these mechanisms begin, of course, with group 1. So group 1 are primarily located post-synaptically and function by stimulating phospholipase C, or as we like to shorten everything down, PLC, to increase levels of diacylglycerol and inositol triphosphate, as well as can also activate a range of downstream effectors, which may be important in the regulation of synaptic plasticity. Now into group 2. Group 2 function also presynaptically to suppress neuronal excitability through the inhibition of a denylate cyclase and are potential novel targets in the treatment of anxiety disorders and even schizophrenia. Now we have group 3. And group 3 functions in the same manner as group 2, but it plays a role in the synaptic remodeling and persistent drug seeking and addiction, exerting their effects via an as yet undefined presynaptic mechanism, possibly also extending to postsynaptic modulation. Nice. Awesome. So we've seen this term in part in this lecture so far, but we're just going to dive a little bit deeper by going first through its structure and then through its function in a subsequent slide. So of course, we're talking about G protein. G protein, also known as guanine nucleotide binding protein, is involved in transmitting signals and function as a molecular switch. Their activity is regulated by factors that control their ability to bind to and hydrolyze guanosine triphosphate to guanosine diphosphate, GTP and GDP, respectively. When they bind GTP, triphosphate, they are turned on. And when they bind GDP, they are switched off. G protein complexes are made up of 3 different subunits. As you can see here, we have alpha, beta, and gamma, with alpha being the most prominent and beta being the least, 26 and 12 respectively there as well. Perfect. Moving along just on schedule. Perfect. Feel free to pause me at any question. Anytime you have any questions or concerns. If not, we'll now move on to some visuals. Incredible. So let us begin with just a quick little note on what we just discussed, our alpha, beta, and gamma. As you can see here, beta takes up the most surface area out of the 3, and the other 2 are pretty closely equivalent in their area as well. And also, you might remember all about that, one of the names being 7 transmembrane, and this is a nice visualization of that because 7 of these transmembrane receptors, allowing for the ebbs and flows of the progression of transfer of energy. But first, let's try to stay a little more focused here. So there are 3 general types of dimers, as you can see in the other diagram. Now that we got over our review, we have our homodimer, which has 1 functional outcome possible, and those are specifically our beta 2 receptors. We have our heterodimer, which we have 2 functional outcomes possible. Differential degree of receptor activation determining, in this case, A over B, or B or A. Matter on whichever one is going to be the dominant in that case. And our third one, being most important for our discussion today, that will be our trimeric RM, where there'll be 6 functional outcomes possible. The differential degree of receptor activation based on rank order of agonist concentration. So it could be ABC, BAC, CAB, etc. You get the point. Translocation of one to the other, moving it around, and you can figure out the next combinations in line. Perfect. And why did I say that the trinumeric would be the focus? Because, as you can see here, GPCR. That's functionally what we'll be dealing with, with this specific receptor. But with that being said, it is nice to go over, review, and explore a little bit of the available connections that we can make along the journey of to educational enlightenment. Or at least that's what I've been told. But very well stated, the G protein complexes are made up of the alpha, beta, gamma subunits, thus as you can see on the side. And the beta and gamma subunits can form a stable dimeric complex referred to as the beta-gamma complex, respectively. Pretty straightforward there. But it's just something to keep in mind as we further on in our studies here. And as you can see here, the 7 transmembrane receptors is a appropriate name because there are 7 membrane receptors and they are transmembrane, not one side or the other. And since we're now dealing with how the receptors themselves and them, in this case being the trimeric receptors, I guess the next progression would naturally be a discussion of the signaling molecules themselves. So just to name a few, these include but aren't exclusive to the biogenic amines, amino acids, ions, lipids, nucleotides, as well as peptides and proteins. Nice. Okay, and here they are, signaling molecules and also the associated physiological roles. So some of the main signaling molecules I did just go over, but let's just go with a few examples as well included in there. So starting with the biogenic amines, some examples of biogenic amines include adrenaline, noradrenaline, dopamine, histamine, and acetylcholine. Onto amino acids and ions, we have glutamine. Of course, calcium is important with signaling, so calcium ion and GABA as well. Next one we have is lipids, such as PAF. We have leukotrienes and anandamine. Other common signaling molecules include peptides and proteins. So continuing on down here, peptides and proteins. Some examples of those would be GnRH, angiotensin, bradykinin, thrombin, bombasin, glucagon, calcitonin, vascular-active intestinal peptides, which some of these should be a little familiar because they were introduced a little earlier. PTH, FSH, LH, and TSH as well. That takes us next to nucleotides, which some of the most prominent are adenosine, adenine, and uridine nucleotides. And there's also others. So some signaling molecules are simply light itself. Some are light sensitive. Odorants, pheromones, and opiates. And on to the physiological roles. As you can see here, we have a list of 6 different roles. The first one being visual sense. So that would be more of the rhodopsin, since it is light sensitive. The sense of smell would be more towards our olfactory receptors. Behavioral and mood regulation would be serotonin, dopamine, GABA, and glutamate. Our immune system activity and inflammation would be like chemokines and histamine receptors, while the ANS transmission would be like our beta um, adren. My apologies. Beta adrenergic receptors, as well as apoptosis. So now that we know a little more about the signaling molecules and physiological roles interconnected with G protein, or more specifically G protein coupled receptors, let's look a little more into its associated functions. Nice. Now we're on to the function of G proteins. G proteins are located within the cell, are activated by GPCRs that span the transmembrane. Thus, the 7 transmembrane. You got it. Inside the cell on the plasma membrane, G protein binds GDP when inactive and GTP when active. When the GPCRs bind to signal molecules, the receptor is activated and it changes shape, thereby allowing it to bind to an inactive G protein. And when this occurs, GTP displaces the GDP, which activates the G protein. The newly activated G protein then migrates along the cell membrane until it binds to adenylyl cyclase, which converts ATP to cyclic AMP, which then in turn leads to the next step in the pathway and generates a cellular response. As I know a lot of circling around, that's the whole point, is eventually all of it leads to some kind of cellular response. After transduction, G protein functions as a GTPase and hydrolyzes the bound GTP, which causes a phosphate group to fall off. This regenerates GDP and inactivates the G protein, causing the cell cycle to repeat, or specifically the cycle to repeat. Awesome. And this is a visualization of part of what we went through. So we have the, let's see, we have our GTP, then goes to GDP, and as you can see, that is on the alpha. It's on the alpha section right here. And then the conversion GTP then causes the ATP to be converted to cyclic AMP, and all of that then causes the cycle to return when that phosphate group that has been dropped off of the GTP has been transferred and released. And I like both of these specifically because they show 2 different ways: a nice little close-up of the membrane and a more outward visualization of it all going through the whole cell itself. But with that being said, there are secondary messenger systems involved in signal transduction, which include the adenylate cyclase cyclic AMP mediated pathway and phospholipase mediated pathway. But that being said, GPCRs can also directly activate the ion channels as well. So here begins our next big step. Going a little deeper into our cyclic AMP pathways. As you've heard, I spoke of two specific ones. We'll be beginning with our adenylyl cyclase pathway, which the cyclic AMP dependent pathway is a G protein coupled receptor triggered signaling cascade used in cell communication. When a GPCR is activated by its extracellular ligand, a conformational change is induced in the receptor that is transmitted to an attached intracellular heteromeric G protein complex. Now we have our GS cyclic AMP dependent pathway. The GS alpha subunit of the stimulated G protein complex exchanges GDP for GTP and is released from the complex. In a cyclic AMP dependent pathway, the activated GS alpha subunit binds to and activates an enzyme called adenylyl cyclase, which in turn catalyzes the conversion of ATP into cyclic AMP. Increases in concentration of the second messenger, cyclic AMP, may lead to the activation of an enzyme called protein kinase A, or pKa. Not to be confused with pKa dealing with hydrogens or hydroxides. This is a enzyme. The pKa enzyme is also known as cyclic AMP dependent enzyme because it gets activated only if cyclic AMP is present. Many different cell responses are mediated by cyclic AMP. These include increases in heart rate, cortisol secretion, and breakdown of glycogen as well as fat. Okay, diving a little deeper, we have the adenylate cyclase signaling system. This pathway can activate enzymes and regulate gene expression. If cyclic AMP dependent pathway is not controlled, it can ultimately lead to hyperproliferation, which may contribute to the development and/or progression of cancer. Alterations in number, structure, or function of receptors will lead to disorder in cellular signal transduction, such as hypersensitivity with upregulation, desensitization with downregulation, and receptor gene mutation. And I liked this picture because it gave a nice little feel for what we were discussing, furthering the showing the inaction of the receptor with the GTP and causing the ATP to convert to the cyclic AMP and thus down the hill of it all, causing our next cellular action, cellular response. And with that being said, we covered quite a bit today. So why not end where we began, with a full scale of looking at the cellular system of GPCR pathway. So we covered just the tip of the proverbial iceberg when it comes to G protein coupled receptors, although we went through a lot. But this picture should be a little less daunting than when we started. We should be able to see some of the order within this chaos that the picture once was perceived. But as I said, we still have a long way to go. So with that being said, feel free to discuss with your partners around you on any studying strategies you may want to impose, because we will be concluding our lesson for today. And as always, feel free to email me with any questions that arise before tomorrow's class, as well as our subsequent laboratory investigations later in that day. So thank you all for paying as much attention as you can with such difficult material, and can't wait to see you all tomorrow. Until then, bye bye, and don't forget to study hard.